So Ann, it's a great pleasure to welcome you to the multi-species coronavirus reading group. You've offered us some really interesting material for discussion. So often when we're reading, scientific articles as scholars in the humanities and social sciences, we're struggling to wrap our head around the, technical details. And it's been really lovely to have these stories told in terms of history, in terms of biography, in terms of travel. So first of all, thank you for making virology accessible to those of us who aren't normally able to grapple with the full technical details. You tell a really compelling story about women in science, a story I think that's not told often enough. It's a story about virology, but I think a story about science more generally. And I know, that's of great concern to members in this group, and we'll probably come to that. But maybe I'll just start with an initial question that comes from the just first couple of pages of your the Language of Life article. And this is a story you're telling about growing up in places that I've also inhabited. My own father worked at the Aberdeen Proving Grounds, trying to clean up the mess that you described there. And now they live really near Fort Detrick. And so part of the story that I'm hearing this is a story of militarism and a story of, you tell the story of Cold War science. But at the same time, you're telling a curiosity-driven narrative, and there's kind of these different forces structuring the science that you report on in your biography. You also talk about a couple of moments where you had the opportunity to commercialize some of your findings. So I was wondering if you could maybe just talk about these different elements. First of all, the curiosity-driven, but as a young person, your curiosity was piqued by a science of chemistry that was about blowing things up. At the same time, later on, you're getting interested in very technical problems relating to single-stranded RNA and replication and these molecular structures and genomic sequences. So I was wondering if we could just start there to talk about these different forces that sort of are structuring the scientific enterprise.
Well, first I have to say, one is obliged to live in the era in which you are born. So we don't get to choose that. You from the time you realize there's a world around you and there are people in it doing things and events that are going on that are beyond your control, you either learn to deal with that or you hide. And, I was born after World War II, and that colored a lot of what was going on in the boomer generation in the US. The attitude of the time was, here we are, the US, we're coming out of this, we're now a world power. But it was also the beginning, and well, we went through the entire Cold War, and that colored everything we did. In grade school, we did nuclear bomb drills. They'd take you out of classroom and make you put your head up against the wall, and hide. And it was like, this was just not even considered abnormal. This was just the way things were. So you deal with it. You simply deal with it. People, born now, youngsters coming through, have different experiences. They have their own traumas. But while you're going through it, you don't think it's a trauma. It just is what it is. I went to college through Vietnam, and it was what it was. So it created opportunities and took away other opportunities relative to what you have now, but you live through it. You go through it. My parents' generation had a different paradigm of the way they grew up and the way they did things. But within any of that context, I think as an individual, you sort of have to find yourself. It's not the era that creates you. You are who you are, and you adapt into that. If I'd been born in India, or if I'd been born in Russia, there are elements of me that would have been the same. I might have spoken a different language and had a different culture or different religions, but there would still be elements of me. And I think the curiosity that I had growing up, I still have today, is something that is just innate. And I hope, if nothing else, that sort of came through that narrative, because it's taken many years for me to recognize that I would have done many of the pathways I took just because of the way I am. And it wouldn't have mattered what the context was. And you apply the same curiosity, you apply the same portions of me to whatever's happening today in the middle of a pandemic, as we did in Vietnam, as we did, during the Cold War. So there's an individuality of, learning about yourself, but you can't change the context in which you are practicing that. That's something that's imposed on you from the outside. And for me, we grew up in the Cold War, and it colored everything. But on the other hand, that was the beginning in many ways of the technology race. And without that, there would not have been educational opportunities for women. Vietnam, misogynism kind of got swept under the rug because there were bigger issues. So sometimes, adversity creates opportunities too, or at least cracks that you can crawl through. And you can spend your life ranting and raving about what could have been, or might have been, or should have been, if only, or you can deal with it. And those are really the opportunities that you have. And when Lynn Enquist asked me to write that article, Archives of, Annual Reviews had just started publishing biographies, and I thought it was rather, he had a lot of foresight to start doing that. He recognized that a lot of the old virologists were starting to die, and it would be useful to record some of this stuff. But they never asked a woman before. And Lynn's a good guy, and I knew him very well. And so when he called me, I said, do you want the PC version? Do you want the version to talk to the young generation and make them all into virologists, or do you want the truth? And he actually paused on that, I will say. And then he said, well, tell us the truth, because we know you're going to do that anyway. So it was actually somewhat cathartic to write that thing in the context that it was written, simply because I'd been through a lot and I wanted to put that on paper. And I've been actually very surprised by the response to it, because most of our youngsters today, they don't know about the Cold War, they don't know about Vietnam, they haven't faced adversity in the same way, and what we face in the U.S. is nothing compared to what many of you other internationals face in your own lives or your own cultural histories. So we have it pretty damn good here, and as did I, and so it was an opportunity to do that, but I've been rather surprised by the response to it.
Thank you. I want to give a shout out to Amina, who's a religious scholar joining us from a microbiology lab in North Carolina, and also Abu Farman, an anthropologist joining from New York City. He's written a book about people who freeze themselves and aspire to live
broad
life.
audience.
Yeah, it's a very interdisciplinary group. And as always, if anyone has a question, feel free to just type it in the chat, unmute yourself, or raise your hand, whatever works. So I want to bring you back with the second question to some of the molecular details. And as you're kind of finding your feet as a graduate student, you're working in a lab that's trying to find the holy grail of protein biochemistry, RNA replicase. So could you bring us back to that moment in the history of virology when you're describing this very elegant system for regulating replicase to capsid ratio, and I think for some of the people on the call today, even what a replicase is or a caspid might be something that needs explaining. But if you could understand how the ribosomes, or sorry, if you could slow down and explain for a second how the ribosomes are translating these RNA elements and getting that ratio of replicase to caspid right, I think that could help us unravel some of the fundamentals to virology that we've been talking around over the last year.
Well, it's hard to go back now with, we're coming out of a pandemic here that basically has been stopped because of an mRNA vaccine that was developed in the course of about 3 months and then disseminated worldwide. And we have the technologies now in our laboratories to do that kind of work almost without thinking about it. If I have a gene of any variety coding for anything, I have the tools in my laboratory to clone those things and isolate that gene, study its biochemistry. And my undergraduates can do this. They come in with that technology, okay? But back in the 70s, when I was in graduate school, you couldn't even determine a sequence. So the sequence of SARS coronavirus 2, once that virus came out in Hunan and it was like, oh my goodness, here's a new virus, what are we going to do with this? That was sequenced in 2 days. And one of those days was spent in isolating the virus material in order to determine the genomic sequence. But back in the 70s, to do a piece of sequence of any variety whatsoever, we simply didn't have the technologies we have today. Paul Caseberg here at Wisconsin worked on a small piece of a plant virus that was only 900 nucleotides. And that took 6 years to do, okay, by the technologies of that time. So to be able to do 30,000 nucleotides of SARS-CoV-2 basically overnight that's the difference in the technologies. So if you wanted to study proteins or DNA codes for RNA, codes for proteins, that's the paradigm that everybody learns in high school biology. But many viruses, instead of using DNA as their genetic material, actually skip that step and make RNA directly. That's their genetic material. And the viruses, like coronaviruses, are basically protein shells on the outside that protect that RNA genetic material. And we worked working on a virus the size of a coronavirus with 30,000 nucleotides would have been unthinkable at the time. It was simply far too big and far too complex to even deal with. In fact, I'm not even sure if coronaviruses were on the radar back in the 70s because they were far, far too big and too complex. So people worked with the much smaller viruses that were much easier to isolate. And many of those were bacteriophages. So there's every cell on Earth, including bacteria and algae, have viruses that infect them. And the fast viruses, the ones that go in and kill things quickly, all for the most part have RNA as their genetic material. They spread very quickly, they divide very quickly, they don't have to go through a DNA phase. So RNA viruses are the bad nasties, basically, of the virology world. And that's true of plants and animals and every life form on Earth. So I've studied, Paul Caseberg, who was my mentor, was working on RNA viruses. And he was working on the tiniest of the RNA viruses, these bacteriophage. And they have only 3,000 nucleotides in their entire genetic makeup. They only code for 3 genes. 3 total genes, whereas a coronavirus is 30,000, it's 10 times bigger, and codes for 17 genes. So it's like if you want to learn about internal combustion, you start with a lawnmower and you take apart a lawnmower, and you figure out what the carburetor does, and take it apart, put it back together again, and then you're ready for something bigger. Then you start maybe working on a Volkswagen, and then maybe you move up to a Ferrari. Okay, but at that time we worked on the little guys. And bacteriophage, just like every other virus, needs a cell that it has to infect. It uses the machinery of the cell to replicate. So the virus will bind to the outside of a bacteria and inject its RNA into the cell. And then that RNA has to make the 3 virus proteins that are needed basically to make new baby viruses. So it has to be able to copy itself. And we don't have no bacteria or mammalian cells have an enzyme that will copy RNA into RNA. They have enzymes that will copy DNA into DNA or DNA into RNA, but not RNA into RNA. So that's a very special kind of enzyme. And again, it's a very simple type of enzyme. So we figured if we could figure out how the viruses did this, we'd then be able to look in a cell and see how the cells do it. So you can isolate bacteriophage. It's quite easy. You infect E. coli. And from a liter of E. coli, you can get, basically a cup full of virus, okay, from which you can extract the genetic material. And there are 3 main genes in bacteriophage. One of them helps kill the cell. Most viruses have that kind of thing. And then there is a tandem cistron. That means 2 genes linked together. One codes for the coat protein on the outside, basically the wrapper for the virus. And then there's a spacer, and then the polymerase gene, which is the core of doing RNA to RNA. And a ribosome will bind to the beginning of the capsid gene and start there. There's an AUG, the first codon of the capsid, and it will read through and start making the capsid protein. And when it gets to the end of that gene, there's a short spacer. And at that point, the ribosome can do one of two things. It can fall off, okay, in which case you only made the one gene, or it can scan through the spacer region and then grab the beginning of the polymerase and make the polymerase gene. And when the virus goes to package the babies, make new virus, you need 60 copies of each of those capsids to make one new particle. So a particle of virus has 60 capsid proteins on the outside, actually 180 of that particular protein, 180, and only one RNA on the inside. So you need 180 times more capsid than you need RNAs to be able to make the babies, right? So the virus has to work out a way to regulate, how do I make 180 of the upstream gene and only one of the downstream gene, which is making the polymerase? Otherwise, I'd make far, far, far too much nucleic acid than I need. And viruses are nothing except efficient in the way they do things. So the way they regulate that is to put a special little RNA structure, a little feature in the RNA in that spacer region. And when the capsid protein is made, it jumps off the ribosome. And if it binds to that spot, it regulates whether or not the ribosome can continue through to the polymerase. So it's a sort of how do I put it, self-regulating mechanism. Because if I have enough capsid around to be able to bind there, obviously I don't need more polymerase. So it's a cute little trick for being able to regulate the ratio. You can use the same ribosome to go through both of them, but it will only go all the way through if there's no protein sticking in that middle piece. And that biology was well known. It had been discovered the year before I entered the laboratory. And the trick was, we didn't want the capsid protein. We knew everything there was to know about the capsid protein because you can study the virus. What we wanted to do was put the polymerase in a tube and be able to study how RNA was made from RNA. How did the virus genome get replicated to be able to make the babies? And we didn't, because that would teach us how these enzymes worked. It would teach us how RNA was made into RNA. And from that, we could also figure out, since all of these enzymes are quite related to each other, how DNA is made into RNA or how DNA is made into DNA. And so in other words, how your cells replicate, how your body is able to replicate and replicate you is the same mechanism that these viruses use, but on a bigger scale. So if we could study the simple enzyme made by these viruses, we could extrapolate for what also happened in the cells in a much, much, much more complex environment. So we wanted to be able to put that stuff in a tube. And in order to do that, we had to figure out how to trick the system into making lots and lots and lots of polymerases without shutting itself off, which is what the virus, of course, wanted to do. And you do that by putting mutations in the capsid gene, and the ribosomes would hit these mutations and stop. And if you put the mutation very early in the capsid, it would make a little piece of capsid, which was too short to bind and shut everything off. But if it was too far away, the ribosome couldn't jump. It couldn't, in other words, if it stops too early, the gap is too big, is a way of looking at it. If it stops too late, then the protein is big enough to repress. So we needed a spot somewhere in the middle, literally like Goldilocks. So you wanted the piece that was just the right size, that the ribosomes could still jump the gap. The gap wasn't too big. It would keep making polymerase, but the protein wasn't big enough to shut it off. And the people in the lab ahead of me had made this wonderful panel of mutations, but they hadn't been able to test them. So my first thing I did in the lab was take the panel of mutations that they had made and test them for their ability to make this polymerase. And lo and behold, there was one in there that was just right, literally like Goldilocks, you know, exactly the right size. And that when we grew that particular virus in the lab, it would make huge amounts of this polymerase, which then we could isolate and study. And it just sometimes things just fall in place the right way. We got exactly the right piece at exactly the right time. And I could spend 4 years studying polymerases. Those of you who are not in science may not recognize this, but almost everything we know about molecular biology is derived from virus studies. And that includes the genetic code, that includes the DNA to RNA to protein paradigm. The first structures of protein were done with viruses. And the reason for that was that back in the 60s and 70s, before we had recombinant engineering, you could put virus in a tube. You could purify it and put it in a tube and study it because you had pure genetic material and pure protein. Trying to isolate something out of a cell, you have all that other cell stuff in there, but viruses are very, very simple. They have only a few components. So if you want to study nucleic acids, if you want to study the genetic code, you want to study how ribosomes work, almost all of that work is founded on early elemental virology. And much of that with bacterioviruses, as Paul Caseberg was doing back in the 70s.
So I see a question from Francisco. And first of all, Anne, thank you. That's, I think, the best explanation of a virus that we've had after more than a year of doing this. You know, we've talked a lot about how, you know, seeing viral particles is, you know, like misleading. You know, it's like the seed of a plant. You don't study trees by looking at their seeds. So we've been thinking a lot about, in the words of John Dupre and Stefan Gudinger, about viruses as living processes rather than, you know, trying to sidestep that dualism between life and non-life. But yeah, you've just made viruses come alive, so thank you.
Well, I've been asked that question 100 times this year of whether viruses are alive. And I think all you need to do is you know, think about COVID to answer that question. They're like, think of them as pollen grains, you know. I mean, it's an entity into itself. It's a capsid on the outside that protects the genetic material during the transfer.
Francisco, do you want to jump in and ask?
Sorry, I interrupted you there.
No, all good.
Thank you, Ben. Well, I have just written a question. I guess it's related to worldviews about virology as a science in the current moment, right? So I come from history and philosophy of science, and so that's why I'm asking you if you think that what we need is to continue going deeper and deeper into molecular genetic mechanistic investigations, or if we should take into account the many other processes that have to do with cells, developmental processes in the sense of Susan Oyama, I suppose, the author of Developmental Systems Theory, and things that are studied by sociologists and anthropologists and other social scientists. What would be your perspective as a molecular virologist on this?
Well, science is no longer either or. When I started out in biochemistry, there were very big distinctions between the physicists and mathematicians who did structures, biochemists who did enzyme kinetics, for example, how do the replicases work, cell biologists who studied the processes in cells, immunologists who studied what happened in the body, and you know, you could walk right up the food chain to the clinical, or the people working in the field, anthropology, looking at, you know, dinosaur bones. And we used to think of all of those as separate entities. But it turns out no matter where you enter that continuum, it is indeed a continuum. And without having been exposed to sociology or anthropology or cell biology or immunology or molecular atomic physics, when I was a student, I would not be able to move up and down that food chain the way I have in my career. And in fact, it is essential for any scientist at any level to have mastery of a great number of those disciplines these days. In my laboratory, for example, I may have physicists and mathematicians working alongside people doing enzyme kinetics, who then hand the project to the cell biologist, who says, aha, this is what that protein does in the cell. And then the immunologist who says, aha, this is what happens when it's displayed on the surface, and the antibodies want to bind it. And then to the pharmaceutical people who are saying, aha, this is the way in which I make that into a vaccine. And then they're standing next to MDs and DVMs who are taking that material and putting it into people and doing the clinical stuff. And we're now involved in the sociology and anthropology, if you will, of where did that gene come from in the first place 50,000 years ago or 100,000 years ago that changed whether or not people are today susceptible to asthma. So you're probably not familiar with our most recent body of work, but we've done the biochemistry of asthma, and it dates back to 150,000 years in human evolution. And we can put the start date on different cultures throughout the world today and where their genealogies came from that led them to be or not be susceptible to particular viruses. So you can't do one without the other, and it's not done in isolation. I've found I can take a biochemist and I can move them up and down the food chain pretty easily. I can teach them physics and I can teach them anthropology. But if I bring people in at other levels, take a cell biologist and try to teach them the physics of a virus particle, they don't have the background and training. So students coming in today have to master a large number of these disciplines. And I'm constantly learning myself, because if you follow any result to its conclusion, that means taking the molecular result, putting it back in the cell, taking the cell, putting it back into the body, taking the body, putting it back into the culture, and putting the culture back into evolution. So you have the way we do things today, because we have access to all of the genetic data and also the anthropological genetic data. You sort of have to be cognizant of this and be able to follow your project all the way up and down the entire continuum of science. So it's no longer I'm a biochemist, okay? It's I'm a scientist, and you got to follow with the technologies and of where the project leads you.
Thank you, Ann.
Thanks. One of the other things I really liked about your article is the way that you kind of laid bare how accidents were a key part of the experimental process. It's often something that's kind of redacted from the published, scientific literature. The large dose of uric acid that led you to make a critical discovery in your career, but then also I found it really interesting how this paper with famous monsters. Where the PI Spiegelman was claiming that he'd found an enzyme that could create life, how that was kind of an artifact of science, and ultimately it sounds like your own findings weren't published, that sort of put his particular experiment in context. So I think that speaks in part just, like there's power relations, like who's getting published and who's not getting published at particular moments of their career. But I think it's also, a philosophical conundrum. How do we think about these fragile artifices of knowledge that are sometimes based on mistakes, and how might new kinds of mistakes produce frame shifts in the sorts of knowledge that we might create?
Well, the accidents that turned out to be fortuitous are the ones you remember. Okay, so let's put that straight first. So it's not like you're constantly doing things wrong. It's that occasionally you do things wrong, and then later you look back at it and you go, oh wow, good thing I messed that up, because that was like, okay. So you remember those and you write them down and you go, oops, that's how it works. Yeah, you can't take a mistake and just let it stop you. You have to move around it. We all make mistakes, and most of them are indeed mistakes, and you back up and you move sideways and do something else. But in terms of those mistakes or the power structure getting in the way of real science, I think that's really not an issue. What it does is it may delay moving forward in the right direction. But if there are papers out there that are, how do I put it? Spiegelman did experiments and he made observations and he published the observations, okay? No problem with that whatsoever. The problem was that his interpretation of what he saw was this is brand new de novo synthesis. This enzyme is creating life, okay? So there was nothing wrong whatsoever with the experiments he did or the observations he made. It was his interpretation that there must be no other explanation other than this is de novo. So it wasn't the experiment, it was the interpretation. And I had done experiments with the enzyme and I knew my enzyme was better and I had an alternate interpretation. Again, my experiments, we had published those here in Wisconsin that the enzyme was contaminated with RNA, but nobody was going to read my results as thoroughly as they were going to read his. On the other hand, people tried to follow up both of those by, oh, wow, if we can create life, I'm going to make this enzyme and I'm going to do it. And they would try to reproduce his results and couldn't do it. And they would try to reproduce my results and they could do it. Okay, so it comes out in the wash eventually. There was nothing wrong with my observations, nothing wrong with his observations. The only dichotomy was the interpretation of that. And it took a while, and that's fine. That's just, you know, power has its structure, right? But you know, eventually things got sorted out in the literature simply because the observations themselves were correct in both cases. Just the interpretation was different. Okay, but yeah, there's a power structure. It still is now. Who would anybody be to argue with that? Anybody in any field here who's listening knows that. You know, depends on who says it, right? But again, remember, it doesn't matter who says it. It doesn't make that correct. It just means that it may take a little longer to correct it in the literature. Somebody somewhere will fix it.
Amina, do you want to chime in? You've got some related comments in the chat.
Oh, I think I've, it was merely a comment that I do think, I also don't think it's by accident that an accident can be appreciated for what it is. I'm grateful that that part of the story isn't hidden. It makes science feel accessible and human. It's not robots that are doing this kind of work. Yeah.
Well, when we published that result about the urea gels, I didn't write in the paper that I put the wrong urea in there. I put in the methods, we used 5 molar urea and this is what the gel looked like. Okay. Not that I wasn't supposed to have used that concentration. It was like, here's the method that we used and by golly, then you get this result. So you know, I would say you cover up your mistakes, but you truthfully tell what happened and then somebody else can reproduce it. People came up to me at meetings and said, well, why'd you ever put that much urea in there? And then you tell them, well, I didn't mean to, but it worked. So in that case, okay, so you publish what you did, but you don't never lay out that, gee, I didn't really mean to do that.
I'm interested also in the idea of monster, like the figure of the monster is something that's very widely used in the humanities and social sciences. You know, everything from we have conferences on Frankenstein and all kinds of other things these days. So can you tell us a little bit more about what the Spiegelman monsters were as conceptualized, even though now we understand that there was an error in interpretation?
Remember this was the, let's see, Spiegelman monsters would have been the 60s, early 60s, late mid 60s, when Saul Spiegelman was experimenting with enzymes, and then we knew about DNA, we knew about RNA, we knew about protein. But the question was, where did the first genetic material come from? How did those first sequences ever arise on Earth? And you know, where did nucleic acids come from? And how did those sequences get there that evolution could begin selecting from? And when the Q beta polymerase was first isolated, and Spiegelman had done some of that very crudely. My enzymes were much better, but I was the generation after him. So I knew how to take what he had done and fix it, so to speak. But so he had very crude enzyme in his preparations, and he would purify it as much as he thought was possible with the techniques at the time. And at that time, it was dogma that you needed to have a template. You needed to have a pre-existing piece of genetic material that the polymerase would copy. Okay, so you would put your genetic material in there, viral RNA, whatever it happened to be, and you would add the nucleotides, which are the substrates that are going to be polymerized into the new progeny, in with the polymerase, and the polymerase with great fidelity would copy the genetic material that was in there. Okay, so he purified his polymerase and did not add back to it any template. He just took what he thought was purified polymerase and just added the nucleotide substrates, the 4 bases, A, C, G, and U, because this is RNA, not DNA. You don't have a T in there. It is uridine instead of thymidine. And he would add the 4 nucleotides into his purified protein. And from purified protein and purified nucleotides, out would come polymerized nucleic acid. And it was about 6S, which means it's about maybe 300 nucleotides in length. And that's what he would get back. So with pure enzyme and no template whatsoever, in his opinion, out would come this piece of genetic material. And every time he did the experiment, he would get back genetic material that had a different sequence. So in other words, every time you did this experiment, you'd come up with a different nucleotide combination. And it was basically making nucleotides, making polynucleotides, making new genes just from purified polymerase alone, plus the substrate, the nucleotides. And so basically he maintained that life on Earth came about because we had there were enzymes or protoenzymes and nucleotides, which of course were in the primordial soup. And it was the enzymes in there that were basically creating sequences and making life, making new genes that could be selected from. So all of that original work on the origin of life, where did it come from? How did life originally figure out how to make polynucleic acids out of the original nucleotides? How did that happen? Here was an enzyme that was doing this de novo, just straight up. Mix the two together and bingo, you'd get a new gene coming out. And when I started working on that polymerase, I had much better technologies than he did when he did his. So I could purify this stuff better. I had better biochemistry. I could characterize the enzyme a little bit better. Again, I was the 2nd generation working in that system. And my mutations allowed me to make much more enzyme. And the more you have, the more you can purify it. If you start with a tiny, tiny little amount, it's very hard to purify. But if you can make lots of the enzyme, you can get rid of the impurities much better because you're starting with more material, okay? So by starting with more material, I could make better stuff and I could make cleaner stuff and I could characterize it. And when I started characterizing it, first thing I looked for was residual template, okay? And it turns out that enzyme binds to pieces of RNA from the genome and it carries it with them through the purification process. And I could detect that signal biochemically. And so I knew very, very well that the enzyme I was making, which was much better than what Spiegelman had used several years before, was actually carrying template with it. And while he was not adding new template, it didn't matter because there was already genetic material in the sample by definition. So the material that he was measuring and his measurements were correct, he'd mix enzyme and the nucleotides and out would come product, right? But I knew that product was actually templated on the residual pieces that were in there because I had done more advanced biochemistry than he was able to do. So his observations indeed were correct, but he extrapolated and said the enzyme has to be doing this de novo. And my work showed that there was contaminants in there that were responsible for this. But my boss at the time, Charlie Weissman, wasn't about to criticize, well, Spiegelman eventually got a Nobel Prize, right? So not for that work, but for his work on phage. So he wasn't about to say, oh, here's a postdoc who says all of this is bunk. Okay, lesson learned. Okay. But nobody makes, and they were called monster. He called them monsters because every sequence that came out, every time he did the experiment, he got a different sequence. As expected, it was templating on random pieces of RNA that were captured by the polymerase.
Melanie, do you want to jump in and ask your question? I think it pretty directly follows from that last answer.
Yeah, sure. Thank you. I really liked the making of story that you shared with us in the beginning, and how, probably unconsciously, by inviting women to write about their biography in a science journal, like the journal invites political commentary, and also like ends up with like keywords like fate in the archive, which is a radical intervention, I think. But I would be keen to, if you could just tell us a bit more about the reactions to the piece, because I really appreciate the honesty, and also how you make very powerful suggestions, like how practically things can be changed, like a process like peer review, for example. Like, have any of those suggestions been taken up or been discussed? Or I'd just be keen to hear a bit more about that. And just thank you so much for writing this piece. And also, I think you should write a full-length biography, just like, as a global history of the 20th, 21st century, these women's biographies that include so many stations across the world. It's just super fascinating. And there were so many parts of it where I thought I would like to read more about like this place and that stage in your life and in your research.
The difficulty with this autobiography was at what level to pitch it, because it's basically pitched at people in virology, graduate students in virology, because there's technical terms in there that don't resonate across the board. And when I showed it to people in my family, for example, they would just blank out on the what's a ribosome bit. So it's very difficult to find a pitch that'll appeal both to, say, graduate students in virology and also the lay public. So I've toyed with the idea of writing something longer. In fact, I have written a large number of short stories. How do I put it? That would augment this, but haven't published. I pitched it towards graduate students because I see a great number of them, and they come in and they, how do I put it? They don't know where the privilege they have today came from. They never went through the Cold War. They never went through Vietnam. For them, it's a bad day when the Wi-Fi is down for half an hour. And if they don't get in the lab they want, it's the first adverse thing that ever happened in their lives, and they think their lives are going to end. And I sort of wanted them to see that it's okay to scrabble around, and it's okay to make mistakes, and it's okay to have some setbacks. As long as you stay more or less on the path, things will eventually happen to you if you really want them to. And so it was basically pitched at that audience, but I didn't want to discourage them, but I wanted them to realize the level of privilege that today's scientists sort of kind of have, okay? The bit about fate I put in there because I'm a scientist and I would be the last person to say I know everything. So that which I don't know, right, you put into this basket called faith or religion or God or whatever, and you say there's a whole lot about life and there's a whole lot about this process I don't understand, but I do understand that it's out there, and so let's just put it there, okay? It's how do I put it? It was cathartic to be able to write this, and I wanted youngsters today to understand that it's okay to fail. It's okay, just stick with it. But on the other hand, don't expect things to be handed to you. If you just go along and wait for somebody else to decide your fate and gift you with a position or tell you what school to go to or tell you what to study, if it doesn't burn in you to do it, you're doing the wrong thing. You'll find your own path if you look inside yourself.
The image of your mom laying in front of the car comes to mind as you drove around her off to your destiny.
She was not happy. She wanted me married and working in a bank, giving her grandchildren, But again, that was the time of Vietnam. Well, and also you have to remember the Sterling Hall bombing was here on campus 2 days beforehand, and that was that was one of the similar to Kent State, one of the defining features of the Vietnam era that basically pulled people back to sanity. It was when we started having, episodes like that of domestic terrorism that really hurt people, that everybody sort of pulled back and went, wait a minute, we got to stop and reanalyze the craziness here. And that was huge on this campus, that Sterling Hall bombing. Before then, everything on this campus was chaos. There were weekly, riots and tear gassings and this kind of thing. And then people got hurt in a bombing, and it was like, wait a minute, this has gone too far. And that was true nationwide. So, from that point forward, there started to be healing. But that was 2 blocks from our building. Army Math Research was across the street. And in fact, the Army ROTC was immediately across the street from the Bach Labs here. And we used to have National Guard with rifles standing on the parapet of this building. We were tear gassed out of our building multiple times that first year because of the riots in the street right in front of our laboratories. And when people started getting hurt, suddenly the craziness stopped or slowed down as sanity, started to come back. So it was an interesting time to live through. And she was, why are you going to Wisconsin to do this when you could be working in a bank here and give me grandchildren?
I think, Abu, do you have a question in the stack?
Yes. Can you hear me?
Yep.
All right, great. Hi, thank you. All this talk of accidents and monsters make me think of mutations and all the talk of variants that we've been exposed to. And I guess I don't know all the technical biology here at all, but I've been sort of trying to wonder, I'm wondering about the variants and how in the RNA replication or RNA virus replication process that you partly described, and it goes way above my head in its details. But then and you talked about, I think, somewhere at the end of that article about at some point discovering all this variability that wasn't supposed to be there. I'm just thinking, how do RNA viruses mutate? And by that, I'm thinking, how do we understand those mutations? Are we thinking that they're simply sort of copying errors, replication errors? Are they strategies for adaptation? And what would be the difference in terms of how we understand viruses if it was one or the other?
Well, that's a very good question. And a large number of people much smarter than me in genetics have spent many, many years working on that. And it turns out that, maybe not surprising to anyone with a religious background, but all of life is sort of random. Okay. And when a virus has RNA as opposed to DNA as its genetic material, DNA is double-stranded. Again, you probably all learned that in high school, that there are 2 strands that are complementary to each other, and they both code essentially. There's a positive strand, and the negative strand has the same information but in the inverse. And when polymerases copy DNA, or when they copy your genome, they move along one strand copying it, and another enzyme moves along the other strand copying it. And whenever they reach a spot where they sort of disagree and they're not sure what nucleotide to put in, they cross-check with the other strand. So in other words, you have a backup mechanism to make sure you're not making mistakes. So when DNA is replicated, the polymerases basically have not only the copy that they're making, but they can cross-check with the other one and make sure they have not made mistakes. And as a result, when your genes replicate, when your cells divide, or you have a baby and the baby is making brand new cells, a new type of life, the fidelity with which that happens is actually quite high, because otherwise you would be giving birth to frogs or dogs who would have much more variation of life. So DNA to DNA copying is actually with very good fidelity. But the viruses that choose to use RNA as their genetic material are primarily single-stranded. They have only one strand of genetic material. And when the polymerase is going from one end to the other, making a progeny strand, it doesn't know when it's made a mistake because it has no other second strand to check it with. It's just copies, and if you make a mistake, it's like typing, you know, without a correction, right? If you hit the wrong key, too bad. It just keep right on going. There's no erase function, right? So the polymerases, and particularly the RNA polymerases that we studied at Q beta, one of the things that came out of that was how frequently do they make mistakes? And it turns out that they make mistakes probably 1 in 500, 1 in 1,000. So that means if you have a piece of genetic material that's only 3,000 long, every single one of them has 3 mistakes, okay? So if I make multiple, no 2 viral RNAs then that are 3,000 long, they will differ from each other by at least 6 nucleotides, 6 mistakes in each, 3 mistakes in each one, right? So you never get a perfect copy of RNA. You always get variable copies, and we call these mutations. So it goes, it's supposed to put in a C, but a U looks very much like a C, and so it just sticks that in, just keeps right on going. There's no way to go back and you know, use whiteout and fix it. You just keep going. And when you've got things like coronaviruses that are 30,000 long, they're making a lot of mistakes. But coronaviruses that get that big actually have new proof checking mechanisms that we're only now beginning to understand. And in viruses that are that big, those proof checking mechanisms that take out bad nucleotides are why a lot of the nucleoside analogs, the anti-drugs we want to use, don't work. The virus fixes those mistakes. So that was a problem with making drugs with coronaviruses. We started to understand that mechanism, and that virus particularly fixes a lot of the mutations. And smaller viruses don't. They don't have a mechanism for doing that. So they're making a lot of mistakes. And as a result, if I have a tube full of viruses, say Q beta viruses, no 2 of them actually have the same sequence. So when we go to infect cells with that pool, some of them will have sequences that are viable and can infect, and others have made mutations that kill the virus, basically. They'll try to infect, but they'll be bad. So natural selection is always looking for sequences to select from. And the viruses, by the means of replication that they have, are providing that variability every time a cell lyses. Every cycle of replication provides variability, and then natural selection, which sequences are good and viable, which ones are not. And so there are no 2 that are the same. We call this quasi-species, semi-species, right? So in a tube of virus, they actually are not genetically identical, unless I convert these days I can genetically engineer viruses and make them all identical. But only the genetically recombinant ones can do that. But in nature, when a virus infects, no 2 of the babies coming out are identical.
Thank you. And you know, my opening question was about militarism, and I'm going to conclude with a question about peace. So with Sacha Gorbaleno, we read also one of his lectures that was oriented towards a popular audience with the open-ended question in the title: "Can we live in peace with viruses?" And you know, as we think about variability, whether it's of you know, the coronavirus or these rhinoviruses or others that you spent your career studying, I was wondering if you could just say a little bit about yeah, the relationship of the virus to its host. You know, one of Sasha's basic points in thinking about SARS-CoV-2 and other coronaviruses is that, you know, the virus that kills its host has reached a dead end. So well, okay, I see you shaking your head. tell us your take on, you know, can we live in peace with viruses?
The last cell on Earth will be killed by the last virus on Earth. I mean, that's a given. that will come true. But from a virus perspective, a newly evolved virus, something like SARS-CoV-2, which it comes out of animals and finds a new niche in a new host niche, an immunologically naive population like humans. We have no pre-existing immunity to that particular serotype. So in other words, everybody is vulnerable. if there's a 13% mortality rate, which is without medical intervention, essentially what SARS-CoV-2 would be, and that virus killed 13 to 20% of all humans on Earth, in terms of evolution, no one would care. Okay, this has happened thousands of times to every form of life on Earth since there has been life on Earth. That kind of mortality is absolutely acceptable in terms of evolution. You know, look at what smallpox did when it was introduced, you know, into South and Central America. That was a 90% mortality. Okay, and you look back now and you go, so what? The survivors are immune, or in fact, Europeans were immune, having survived multiple 90% mortality epidemics. Okay, so everything we know about immunology, everything we know, your entire immune system is a sum total of evolution fighting against exactly these kinds of events over millions of years. Okay, so that we have an innate immune system, that we have an active B cell and T cell immune system, is because these kinds of events have occurred over and over and over and over, and will continue to occur over and over and over. Okay, this is just the latest. What is amazing about this one is we actually now have medical intervention, which takes that 13 to 20% native mortality and drops it down to 2%. Okay, and we consider that socially unacceptable. But from a biological perspective, oh my God, you know, that you can, within 6 months of that virus coming out and starting to kill people, you can start to immunize and protect populations. This is unheard of in the history of evolution. So, you know, you can rant and rail, but the investment that was made in science that allowed that to happen, you know, saved millions of lives, which we consider precious. But in an evolutionary standpoint, if this SARS-CoV-2 had come out 500 years ago, it would have had a 20% mortality, and now that virus would be endemic in the population, and we wouldn't even be thinking about it. So it's a question of timing. Okay, so if you think this is the last one, you're absolutely mistaken. Okay, and viruses, as long as there is a continuation of the host, what do you care, right? Red tide. I mean, and instead of putting it in human perspective, let's look at things like you all know about algae blooms. You know, red tide, and you have these algae blooms in rivers and you know, from pollution and waste. Those are a type of algae or bacteria will start to proliferate. Those go away because a virus comes in that is adapted to that particular thing and kills every one of them. Every one of them. All right. Those red tides go away because virus lyses the cells. Okay. Now, that's not to say that that's the last ever of a red tide, but it's going to take a little while for another one to come back up that's, you know, not going to be susceptible to that particular virus. So viruses with close to 100% mortality, this is a natural part of natural selection over and over and over since there has been life on this earth, and it will continue as long as there is life on this earth.
So on that dark and hopeful note, we will conclude the conversation today. So thank you so much, Ann, for sharing your biography with us, for sharing your thoughts, helping us understand some of the basic virology, but also, you know, the social context in which virology takes place. It is a very important and enlightening conversation. I'll do a brief plug for next week. So on September 13th, next Monday, we have a PhD student from MIT, a historian of science, Rijul Kochar, who's talking about the history of phage research in India. Actually, before, you know, phages were isolated as a thing that you could, you know, identify with advanced molecular techniques or an electron microscope. There were early colonial scientists going out and trying to figure out why the river Ganges was being seen as a medicinal curative source. So the secret may well be phage. So thank you, Anne. A big round of applause, virtual applause, and yeah, hope to see you in another Zoom room or even in real life sometime soon.
The American Society for Virology is coming to Madison next July.
Oh, it is. Okay. Well, we should be there.
In person.
In person. Thanks, Anne.
Hope to see you there. Thank you.
Okay.
Thanks,
Have a
Anne.
great day.
Great.
Have a great day. Bye-bye.
Take care.